Okay, hi everyone. Thanks for coming out today. As Rhiannon mentioned, I'm Soumya. I'm a graduate student in the Zhang lab, and I'm going to discuss some of the technology surrounding RNA targeting with CRISPR systems. And it looks like the slides are a little cut off at the bottom, so I apologize if any of the figures are a little less visible than we would have liked, but I'll try to work through the technical difficulties. Okay, so CRISPR systems basically come in like two main flavors, which are class 1, which are multi-effector systems, and class 2, which are kind of the ones that are more widely developed as nucleic acid targeting technologies, and probably the ones that you guys are most familiar with. So we just had two great talks about applications of Cas9 and other genome editing CRISPR systems. And so today I'm just going to discuss Cas13, which is also a single effector system, but targets single-stranded RNA instead of DNA. So in my talk, I just want to discuss a little bit about kind of like the biology and mechanisms of Cas13 with a specific focus on the components that have been really important in being able to develop it as a technology. And then I also want to just mention some of the different. Oh, great. Thank you. So then second, I just want to cover some of the kind of interesting and creative ways that people have been able to apply this technology or apply this system to target RNA. And finally, I'll discuss a little bit of some like very basic experimental designs that you can follow to be able to use Cas13 as a programmable RNA targeting protein. Okay, so first, I'll just start with some of the biology of Cas13. And that's a little too far. Okay, so Cas13 is an RNA-guided RNA targeting nuclease. So basically what this means is that it's able to cleave RNA. And so as you can see here, it functions kind of similarly to Cas9, where you have a spacer that is able to target a transcript of interest. And you can see here that when a specific targeting spacer is present along with the protein, you're able to see cleavage of a specific target RNA of interest. There's a lot of different Cas13 subtypes that have been discovered, and so this is just kind of an overview of those that are now available. And there's 4 main subtypes. They come with kind of different associated proteins. So for example, Cas13b is known to be associated with 2 proteins, CSX27 and 28, which are able to regulate its function by enhancing or inhibiting it a little bit. More recently, Cas13d was discovered last year, and this is a protein that's associated with these YWL domain-containing proteins. And kind of one of the interesting things is that there's a kind of wide variety of sizes of these different effector proteins, and so that can maybe enable different and diverse applications. The one main conserved feature that these Cas13 proteins have is this HepN domain. Apologies for the, okay, there it goes. So as I mentioned, the HepN domain is a conserved feature of all Cas13 proteins, and this is the domain that mediates RNA cleavage. And so it's found to be highly conserved among all different types of Cas13s. It consists of this arginine and histidine motif that are separated by 4 residues, and this mediates the catalytic activity of Cas13s. So some recent results from our lab have described the crystal structure of Cas13b. And so this adds on to the kind of existing knowledge of the structural properties of Cas13 proteins, along with the structure of Cas13a and Cas13d. And so as you can see, they're a little bit structurally diverse, but the HepN domains are largely located at the termini of the protein. And specifically, Cas13b is known to be a little structurally different than these other 2 subtypes. So one interesting property of Cas13 that at first might not seem like very ideal, but has actually been leveraged into some interesting applications, which I'll discuss later, is that Cas13 is able to exhibit collateral activity of RNAs in vitro and also in bacterial cells. And so essentially what this means is that, as demonstrated by this experiment, if the target RNA along with a guide RNA targeting the particular transcript of interest is present, you will actually also see RNase activity acting on other transcripts that are not specifically being targeted in the sample that you have. So here, this collateral RNA is the one that's labeled, and you can see that when the target RNA is present in the sample, you also are seeing activity that is present on these non-targeted, or transcripts that are not specifically being targeted. The last feature of Cas13 that I want to highlight is that it's actually able to process its own CRISPR array, and more specifically, that activity is not necessarily mediated by the two HepN domains that I discussed earlier. And so this is kind of an important technology feature because you're able to make catalytically inactive Cas13s that are no longer exhibiting RNase activity, but still be able to deliver multiple guide RNAs at once, and that allows for multiplex targeting of many transcripts in a cell. And so both the collateral activity and this multiplex ability are both important for kind of downstream application of this. So just kind of like a broad overview summary, we've kind of seen that Cas13 is a type 6 CRISPR system, and that's able to process its own pre-CRISPR RNAs into mature CRISPR RNAs that can be used for targeting a transcript of interest. It's able to exhibit target-specific cleavage, and additionally, when the target RNA is present in the sample, in certain contexts, we can also see collateral cleavage of non-targeted or RNAs that are not specifically being targeted by your guide RNA. So kind of like the more part that people are generally more interested in is like, why are we really interested in being able to do programmable RNA targeting? And what applications have people developed using Cas13 for this purpose? So there's a lot of like interesting things that we can kind of imagine doing if you were able to programmably target a particular transcript in the complex environment of a cell. So for example, we might imagine being able to enhance or repress translation, also maybe modulate splicing through exon inclusion or exon skipping of particular exons. If we could bind a particular transcript of interest, we can assess the localization of that transcript or possibly even alter its localization. So Cas13 is also a nuclease, and an easy thing that you can imagine is being able to degrade that transcript specifically or possibly block its function in some way. Another application that is not on this slide but that I want to mention is that a variety of chemical modifications to RNA have been discovered. So Cas13 is one way that you can maybe programmably bind a transcript of interest and enact a modification function using a fused catalytic domain. And so kind of unsurprisingly, a lot of people have, since the characterization of this, and since this protein has been characterized and the activity has been demonstrated, people have taken it and done a lot of really creative things to apply the programmable RNA targeting function of Cas13. And I just want to kind of highlight some of the different applications that are out there and just provide a very broad overview of some of the different things that you would be able to do using the programmable RNA targeting capacity of Cas13. So as I mentioned, kind of one, probably the most obvious thing that you might think to do is degrade or cleave a transcript of interest. And so in this example, Cas13 is expressed alongside a CRISPR RNA targeting a transcript of interest. And in this particular example, it's targeting the Gaussian luciferase gene. And so when all these components are delivered to a mammalian cell, we can find that we're able to see knockdown of this G luciferase in the target cells. And again, there's a variety of different orthologs of Cas13, and they kind of mediate knockdown and targeting or cleavage at a variety of different efficiencies, but you can see that there are some for which it's very robust, and there might be different applications for which a different level of knockdown would be necessary. Additionally, just assessing the specificity of this, we've found that Cas13 is actually able to quite specifically knock down certain transcripts of interest compared to some comparable technologies such as shRNAs. So in a lot of contexts, though, it's not necessarily helpful to just cleave your RNA of interest. Most of the applications that I'm going to discuss actually involve a catalytically inactivated Cas13, and that's easily achievable by just mutating those catalytic residues of the heparin domain that I mentioned previously. So the first one that I want to mention is being able to image RNA and image specific transcripts and their localization within a cell. So this system, which was also developed in our lab, is the fusion of a catalytically inactivated Cas13 protein that's fused to a GFP for imaging, as well as this zinc finger crab inhibitory domain that allows for negative regulation of this protein in the target cells. And it's been shown that using this protein, you're able to, it's a little hard to see, I guess, with the sliding, but you're able to specifically visualize the localization of a transcript of interest, in this case, the ActB transcript in a cell, but only when the target RNA or target guide RNA is present. So in this non-targeting control, we can see that, or if you can see that the kind of red fluorescence is not really co-localized with the Cas13. So this is kind of an interesting way to be able to specifically bind and visualize where transcripts are localized in cells. So in the first talk, Lindy mentioned DNA-based editing. Comparable technologies have also been invented for site-specifically modifying bases in RNA. So a Cas13 kind of mediated version of this is the Cas13, a catalytically inactivated Cas13b protein that's been fused to the deaminase domain of the adenosine deaminase acting on RNA, or ADAR. So the Cas13 mediates the specific transcript targeting function, and the ADAR mediates the catalytic conversion of adenosine to inosine, which is read out as guanosine by the ribosome. So this is a way to do site-specific modification of A's to G's at the RNA level instead of DNA level. And kind of different to how DNA-based editors work, because ADAR naturally acts on double-stranded RNA, we are also able to specify the exact target adenosine that's being deaminated within the spacer window. And we can do that by including this cytidine mismatch across from the adenosine of interest. And that allows for focusing of the catalytic activity of the ADAR protein to the specific adenosine that you're trying to deaminate. And so results from our lab have shown that this process is quite efficient. And though this kind of shows that you can maybe mediate like 30% editing, we've seen results up to 90% editing on certain transcripts of interest. Another really interesting technology that was developed within the last year is being able to modulate translation using Cas13 binding. And so in this case, the authors of this paper were trying to study kind of like the translational regulatory function of these two proteins, YTHDF1 and YTHDF2, which are known to bind methyl-6-adenosine in RNA to recruit different factors that affect translation. And so what they were also able to develop in the course of this work was a kind of generalized framework for possibly being able to modulate translation of particular transcripts. And they've done this by targeting the Cas13 YTHDF fusion to, in this case, a firefly luciferase. And what they've seen is that, so in the case of YTHDF1, they're able to improve translation relative to a non-targeting control, which is consistent with what was known about this protein before. Similarly, for YTHDF2, they are able to repress translation, again, consistent with what was known about that protein. And this could be really important for being able to modulate translation without affecting transcription of a specific gene. So that is an interesting way to kind of perturb translation at a more or expression of a given protein at a more downstream point than knockouts or knocking down the RNA, for example. So if any of you attended the workshop last year, you might remember that Silvana Konerman gave a talk on Cas13D. Another kind of interesting way that Cas13 has been applied is to be able to modulate splicing by exon exclusion. And so in this technology, what they did was they targeted a variety of sites in this reporter transcript that were known to regulate the splicing or inclusion of this exon. And what they saw was that just by targeting the catalytically inactivated Cas13 to those sites, they're able to influence the exclusion or inclusion of that exon. And kind of harkening back to the multiplex targeting that I mentioned before, what they found was that this process was actually most efficient when they're able to target all 4 of the sites at once, which is a function that's really enabled by the ability to just deliver an entire array of guide RNAs that can target many sites with one system. And what they also found is that fusing the inactivated Cas13D to the HNRNP-A1 splicing factor, they're able to even enhance that a little bit and achieve close to 90% exclusion of a given exon. And so this could be an interesting avenue for therapeutics, as there's a lot of diseases that are caused by inclusion or exclusion of, or aberrant inclusion and exclusion of specific exons. The last technology an application that I want to discuss is not an in vivo technology, but actually an in vitro one that was also developed in our lab called Sherlock. And there's been many updates to this kind of recently, so I will give like a very basic overview of how it works. And again, this is an in vitro reaction that's used for detection of specific RNAs in a complex sample. And this actually takes advantage of the collateral RNase activity that I kind of mentioned before when describing Cas13. And so in this system, you have your Cas13 protein along with a guide RNA targeting a transcript of interest, as well as this reporter, which can be collaterally cleaved by the Cas13 as long as the desired target is present in the sample. And so when that happens, you'll be able to detect the presence or absence of a specific transcript within a more complex mixture. And this technology has been leveraged as a disease detection platform. The other interesting thing is that, which I did not mention before, but I'll touch on now, is that Cas13 has a preference for cleaving at certain dinucleotides, and different orthologs exhibit different dinucleotide cleavage preferences. And so that property has also been kind of applied in this setting to enable multiplex detection of a variety of different target RNAs within one sample. And as you can see, it's actually quite robust. So you're able to specifically detect certain subsets of different transcripts in kind of more complex sample mixtures. And so if you're interested in kind of different applications of RNA targeting with Cas13 or with other modalities, I would recommend definitely checking out a couple of these talks that will be happening tomorrow. We'll have Gayle Mandel from the Oregon Health and Science University, and directly followed by Jonathan from here at the Zhang lab, talking about some like really exciting applications of RNA targeting. Okay, so I'll spend the last few minutes just finishing up kind of with some like very general overviews of how we think about designing RNA targeting experiments using Cas13. And I'll just provide like 2 examples of this process. So first, we'll just discuss RNA targeting in mammalian cells. So the two main components that you would basically need for doing this are the expression plasmids for Cas13 and as well as the CRISPR RNA. And the wonderful people at Adgene have actually compiled a really great database of RNA targeting CRISPR plasmids that have all of the plasmids for all of the applications that I've discussed compiled into one place, and that can be really helpful for kind of deciding what expression plasmids are needed for your specific application. Most of the CRISPR RNA expression plasmids are just backbones that you can use Golden Gate cloning to clone your own spacers into, and so these spacers are very easily designed by just expressing the sequence that's the complementary to your target of interest. And if you're interested in doing RNA-based editing, you simply just have to include a cytidine mismatch across from the adenosine of interest that you're trying to edit. So the design is very simple, and because Cas13 RNA targeting really only requires these two components, they're the only things that you really need to enact this function in any cell type of interest. So again, we just clone the only variable component that is kind of specific to different applications is the spacer in the CRISPR RNA, and this can be cloned again just by Golden Gate cloning into an expression plasmid of interest. So commonly, we just transfect these plasmids into mammalian cells using lipofectamine. As Julia discussed, there's a variety of ways that you can deliver your CRISPR expression plasmids to cells. For non-dividing cells, we found that AAV works well also. And depending on your application, you might read out activity differently, and there's a wide variety of ways that you can imagine doing this. So some examples for implementing this for RNA knockdown, possibly a little excessive, but depending on your application, might be useful, is to do RNA-seq on your sample to determine knockdown efficiency of a particular transcript that you're interested in. Maybe a slightly cheaper alternative to that would be to qPCR your transcript in order to kind of assess the expression of that transcript. And something that I have personally found to be very simple and easy to use is just to target a reporter and just read out by looking at reporter activity. So the second example that I'll discuss is in vitro detection with Sherlock. And again, there's been a lot of different advances to this technology over the years, but I'll just kind of describe like the very most simple, basic experimental workflow. So to go into a little more detail, how this works is you have a complex sample mixture of interest. It could be both DNA or RNA. And by using recombinase polymerase amplification, which is an isothermal amplification procedure, you can amplify this sample and then also use T7 transcription to transcribe it into RNA, which is the substrate for Cas13. Additionally, then by inclusion of the Cas13 as well as a guide RNA targeting the transcript of interest and your fluorescent reporter, you can detect cleavage of that fluorescent reporter in order to, by assessing the fluorescence of your sample. So again, as I discussed, designing CRISPR RNAs using Cas13 is very straightforward. You just design a sequence target that is complementary to your transcript of interest. And specifically, in studying Cas13, we found that 28 nucleotides, sorry, that part I think was a typo, but 28 nucleotide spacers are what are often found in natural systems, and this works well for this application. Additionally, different Cas13 subtypes also have slightly different topologies of the CRISPR RNA, and so it's important to remember that when you're designing in vitro templates for CRISPR RNA expression. So subtypes A and C as well as D have the direct repeat 5' to the spacer, whereas Cas13b, in Cas13b, the direct repeat is on the 3' end of the spacer sequence. So you can either order these as RNAs. Probably the more economical version of that would be to just use in vitro transcription to transcribe your CRISPR RNA. And this can be done by just including a T7 promoter upstream of the CRISPR RNA and using a commercial kit to do the in vitro transcription reaction. And finally, a lot of the reagents that are required for doing Sherlock are actually commercially available. So again, the T7 transcription is a commercially available kit. I believe you can buy them from NEB, amongst others. The recombinase polymerase amplification is also commercially available. And finally, of course, you will need to include the reporter as well as the Cas13 and your in vitro transcribed or RNA, CRISPR RNA that targets your transcript of interest. OK, great. So that was just kind of a very broad overview of the different applications of Cas13 and programmable RNA targeting technologies. And the timing might be slightly off now, but I just kind of want to plug some of the breakout sessions that we'll be having after the coffee break, I believe. And to find these, you can just follow the signs in the main lobby in order to find the ones that you're interested in. So we'll have 3 sessions that are staffed by members of the Zhang lab, one on Cas13, as well as a clinic on Cas9 and Cas12 and other genome targeting technologies. We'll also have a session on CRISPR screening. So if you have any burning questions for Julia, you can ask them to her there. We're also really happy to welcome back Lin Wu from the Harvard Genome Modification Facility to discuss creating mouse models using CRISPR. And finally, this year we have Promega at the breakout sessions to kind of advertise some of their new CRISPR protein tagging technology that they've developed. And with that, I just want to acknowledge all the members of the Zhang lab, particularly Feng, who kindly supports all of the work that we do, as well as Rhiannon and her co-organizers for putting together a great workshop for us. And thanks to all of you for attending the workshop and for your attention. And I think I'm happy to take any general questions, but maybe in the interest of time, you're also welcome to join us at the Cas13 and RNA targeting breakout session if you have any kind of more technical or specific questions.